Hello, and welcome to the Clinical Care Options Oncology Podcast. I'm your host, Tim Quill. Today's episode features expert insight on key new data in lymphomas and CLL in the virtual 2020 ASH annual meeting. This episode is part of Clinical Care Options' broader overall conference coverage from this meeting. During this podcast, Dr. Jeremy Abramson from the Harvard Medical School and Massachusetts General Hospital in Boston, Massachusetts, discusses updated results from key trials with approved and investigational targeted therapeutics, CAR T-cell therapies, and bispecific antibodies for CLL and lymphomas. For more information on Dr. Abramson, along with a link to CCO's complete ASH 2020 conference coverage, including downloadable slide sets, please visit the show notes for this episode. Now let's get started and hear what the expert has to say on these important topics.
So we had some exciting data emerge in chronic lymphocytic leukemia at the most recent ASH meeting. That included long-term follow-up on two pivotal studies evaluating the role of venetoclax and anti-CD20 monoclonal antibodies in both previously untreated and relapsed disease. We have 52-month follow-up of the CLL-14 trial, which evaluated venetoclax obinutuzumab versus chlorambucil obinutuzumab, each given for 1 year of fixed-duration therapy in previously untreated CLL. With this long-term follow-up data, the 4-year progression-free survival was 74% for venetoclax obinutuzumab compared to only 35% in the control arm. And these exciting data demonstrate durable remissions for fixed-duration therapy of venetoclax obinutuzumab in previously untreated CLL. The majority of those patients achieved MRD undetectability, and not surprisingly, patients who achieved MRD undetectability did have a longer progression-free survival. The other long-term follow-up study was the Murano study of venetoclax rituximab, administered for 2 years as compared to the control arm of bendamustine rituximab, administered for the standard 6-month course in patients with relapse or refractory CLL. These data were reported now at a median follow-up of 5 years and demonstrated a median progression-free survival of 54 months for venetoclax rituximab compared to only 17 months in the control arm. If you look at the 5-year point, 38% of patients with venetoclax rituximab remained progression-free in the relapse refractory setting, and the 5-year overall survival for the experimental arm was 82%. So these two abstracts demonstrate very encouraging durability of remissions for venetoclax combined with either obinutuzumab in the upfront setting or rituximab in the relapse refractory setting for patients with chronic lymphocytic leukemia. Another exciting abstract was the evaluation of a novel non-covalent BTK inhibitor called LOXO-305, which was evaluated in the phase 1/2 Bruin study, predominantly in CLL and other non-Hodgkin lymphoma subtypes. What's notable about LOXO-305 is that unlike the other approved BTK inhibitors currently, this is a non-covalent agent and also has activity against patients with cysteine 481 BTK mutations, which renders resistance to the other BTK inhibitors such as ibrutinib. The study evaluated 170 patients with a median of 3 prior therapies. 86% of those patients had a prior BTK inhibitor, 2/3 of whom stopped because of refractoriness to the BTK inhibitor. LOXO-305 was very well tolerated without significantly increased risks of bleeding, bruising, arthralgias, hypertension, or atrial fibrillation, toxicities that we consider with the first-generation BTK inhibitors. And the overall response rate in this relapse refractory population was very encouraging at 63%, and encouraging no difference in response rate based on patients who've previously been exposed to a BTK inhibitor or not. And 94% of patients had ongoing response at a median follow-up of 6 months, so additional follow-up is certainly needed to assess for durability. In other subsets of this trial, among 56 patients with mantle cell lymphoma, also encouraging responses were seen with an overall response rate of 52% and a CR rate of 25%, with 83% of patients in ongoing response at 6 months. And in smaller subgroups, Encouraging responses were seen in patients with Waldenstrom's macroglobulinemia, marginal zone lymphoma, Richter's transformation, follicular lymphoma, and diffuse large B-cell lymphoma. So I think LOXO-305 is an exciting next-generation BTK inhibitor with a highly targeted kinome, which minimizes off-target toxicity and overcomes resistance to the BTK cysteine 481 mutation. And I look forward to more data with that agent in the near future. Looking beyond CLL, we saw exciting data for CAR T cells in indolent non-Hodgkin lymphomas. There were 2 trials reported, including the updated data from the pivotal ZUMA-1 study of axicabtagene ciloleucel, or axi-cel, in patients with relapse-refractory follicular or marginal zone lymphomas. This study included 146 evaluable patients who'd had a median of 3 prior lines of therapy. The overall response rate was a remarkable 92%, with 76% of patients achieving a complete remission. These remissions appear durable, with a 1-year progression-free survival of 74%, though in an indolent lymphoma, obviously much longer-term follow-up will be required. Axi-cel is a more toxic CAR T-cell, and cytokine release syndrome and neurologic events were seen in 82% and 60% of patients respectively, with severe CRS or neurologic events seen in 7 and 19% of patients respectively, though these were largely manageable and reversible. We also saw the first data of Tysogen Lechlucel in relapse-refractory indolent B-cell lymphomas from the ILARA study. Among 52 evaluable patients reported on this interim analysis, the overall response rate was 83%, with 2/3 of patients achieving a complete remission and 69% of responses ongoing at a median follow-up of 10 months. Once again, longer follow-up is clearly required. This 4-1BB co-stimulated CAR T-cell does have lower rates of CRS and neurotoxicity, which occurred in 49% and 9% respectively, and were almost entirely low grade. We also saw CAR T-cell data emerge in both CLL and mantle cell lymphoma. Lymphocyte naralucil was reported in an interim evaluation of 23 patients treated with relapse-refractory CLL and a median of 4 prior lines of therapy, 100% of whom had had a prior BTK inhibitor, and nearly half had had a prior venetoclax exposure as well. The overall response rate in this high-risk CLL population was 82%, with 46% of patients having a complete remission by IWCLL criteria, though importantly, more than 2/3 of patients achieved undetectable MRD in the peripheral blood, with a median duration of response that has not been reached, and a progression-free survival of 50% at 1 year. Long-term follow-up in greater numbers is clearly required. Liso-cel was also reported in the interim evaluation of the Transcend Mantle Cell study of 33 patients who were also heavily pretreated, and nearly 90% had had a prior BTK inhibitor. In this population, the overall response rate was 84% and a CR rate of 64%. So very exciting CAR T-cell data here across multiple histologies in B-cell non-Hodgkin lymphoma, including low-grade B-cell lymphomas, follicular lymphoma, and marginal zone lymphoma, as well as chronic lymphocytic leukemia and mantle cell lymphoma. I think the other major story were the anti-CD20, CD3 bispecific antibodies, which are another attempt at active immunotherapy, but here using an off-the-shelf product as opposed to an engineered cellular therapy that takes time and resources to manufacture. One of these reported was odronextamab, which was studied in a 136-patient study, which included both diffuse large B-cell lymphoma and follicular lymphoma patients with a median of 3 prior lines of therapy. The efficacy in follicular lymphoma was remarkable, with an overall response rate of 90% and a complete remission rate of 70%. In patients with DLBCL, the overall response rate was 40%, with 1/3 of patients achieving a complete remission. There were responses seen in patients who had had prior CAR T-cell therapy, which is notable because these are patients who are difficult to salvage in the relapse refractory setting. The incidence of CRS was 61%, but most of this was low grade and diminished over time, and the durability of these responses was encouraging, though at limited follow-up, so longer-term follow-up is required. Another CD20-CD3 bispecific reported was epcoritamab, which notably is a subcutaneous injection as opposed to intravenous. 68 patients were studied in a phase 1/2 study, including 46 patients with DLBCL and 12 patients with follicular lymphoma, again with a median of 3 prior lines of therapy. In the small follicular lymphoma subset, the overall response rate was 87%, with 53% of patients achieving a CR. And in patients with relapse refractory DLBCL, the overall response rate was a very encouraging 76%, with a complete remission rate of 48%. CRS occurred in 59% of patients on this study, but all of it was grade 1 to 2. And the durability does appear encouraging, though again at limited follow-up, which will require additional time. The third bispecific antibody reported was glofitamab in a 52-person phase 1 study, again in diffuse large B-cell lymphoma and follicular lymphoma with a median of 3 prior lines of therapy. Among all treated patients, the overall response rate was 64%, with 54% of patients achieving a complete remission. And notably, these response rates were quite similar in both the aggressive and indolent histologies. The incidence of CRS was 64%, but decreased with ongoing dosing, and as with the prior products mentioned, encouraging durability at limited follow-up, which will require updating. Then finally, mosunetuzumab is a bispecific anti-CD20-CD3 antibody previously reported in diffuse large B-cell lymphoma and updated in this meeting in relapse refractory follicular lymphoma. Among those 62 follicular lymphoma patients with a median of 3 prior lines of therapy, the response rate was 68%, with a CR rate of 52% and an encouraging median duration of response of 20 months. Notably, this was associated with a low risk of cytokine release syndrome at 18%, all of which was low grade. And so the bispecific antibodies are showing encouraging responses in both diffuse large B-cell lymphoma and follicular lymphoma, with very manageable and encouraging toxicity, which will represent an appealing off-the-shelf product for relapse refractory patients. The last targeted therapy I'll mention for non-Hodgkin lymphoma is loncastuximab tesirine, which is an anti-CD19 antibody drug conjugate. This was studied in a pivotal study, which produced an overall response rate in relapse refractory diffuse large B-cell lymphoma of 48%, with a CR rate of 25% and a median duration of response of 13 months. This was associated in the overall population of a median progression-free survival of 5 months. The drug was generally well tolerated, also represents an off-the-shelf product, and given that this is a pivotal study, it is expected to lead to FDA approval, providing us an additional targeted therapy directed at CD19 in patients with relapse refractory diffuse large B-cell lymphoma. Turning our attention to classical Hodgkin lymphoma, obviously we cure the vast majority of patients with Hodgkin lymphoma with initial treatment. And this is a disease, of course, that predominantly affects younger adults. But it also occurs in elderly patients, and we've long recognized that standard chemotherapy for classical Hodgkin lymphoma has a high rate of toxicity, including treatment-related mortality in elderly patients. This has led to investigation of targeted therapy with minimizing chemotherapy in patients, in elderly patients in need of initial therapy for Hodgkin lymphoma. This initially led to an evaluation of brentuximab vedotin, the anti-CD30 antibody drug conjugate, which is approved in relapse refractory disease or in combination with AVD for advanced stage upfront disease. And when given as a single agent in 26 patients, brentuximab alone produced a response rate of 92% and a CR rate of 72%. Unfortunately, I'm dictating a note. Unfortunately, though this agent had encouraging overall and complete responses, the median duration of response was relatively brief at 9 months. This led to several cohorts evaluating doublets in combination with brentuximab vedotin. The first doublet was brentuximab in combination with dacarbazine. 20 patients were studied in this subgroup, which produced an overall response rate of 100% and a 68% complete remission rate. Most notably, the median duration of response was 45 months, And so this elderly population with a median age of 74 had very encouraging not only response, but durability of those responses using this well-tolerated doublet of brentuximab dacarbazine. Brentuximab was also studied in 20 patients in combination with bendamustine, which also produced a high overall response rate of 100% and an encouraging median duration of response of 39 months. However, this was a more toxic combination, including neutropenic fever and infections, as well as neuropathy and asthenia, and 60% of patients had to discontinue treatment due to adverse events, and so this arm was not recommended moving forward. The last arm that was studied, and the one with the least follow-up, is brentuximab vedotin plus the immune checkpoint inhibitor nivolumab. BV-Nivo was studied in 21 patients with an overall response rate of 95% and a CR rate of 79%. The median duration of response has not yet been reached in this subset. However, the median follow-up is only 28 months. And so I think we're going to need much longer follow-up to see how this compares to, say, the brentuximab dacarbazine combination. I think these data do provide some encouraging evidence for how to treat our elderly patients with untreated classical Hodgkin lymphoma. For this truly elderly population with a median age of 74, brentuximab plus dacarbazine produced a very encouraging response rate with durable remissions and very manageable and acceptable toxicity. We await further follow-up data for the brentuximab nivolumab combination, which also has very encouraging early results.
Thank you very much, Dr. Abramson, and thanks to you, the listeners, for joining us. As a reminder, to view the full ASH 2020 conference coverage program from the Clinical Care Options website, and to download summary slide sets of the different studies associated with this discussion, please click on the link in the show notes. As always, thanks for listening.